So I would like to present our next lecturer, PhD, Professor of Clinical Pharmacy and Computational Biology in the University of Southern California in the USA, Serghei MANGUL, with a lecture concerning the use of bioinformatics for diverse population studies of adaptive immunity. Please, doctor.
Yes, thanks a lot. So really excited to be here and see some bright faces on that part of the screen. So let me try to share my screen. Let me see how I think like this.
We're waiting for your full screen.
Yes. Can you see my screen?
Yes.
Great. So yeah, so again, excited to be here. So if I think question-wise, you can ask during my talk, that's totally fine, in the end of the talk, and I will try to spend some time explaining things. So indeed, I would like to talk about bioinformatics and bioinformatics applied to adaptive immunity in particular. Before doing that, I would like to set up the ground and talk about the way I see the role of bioinformatics in general, biomedical research, and in particular in to study the adaptive immune system. So if you think about the bioinformatics, one way, at least to think about that, to think that it's a frontier between noisy and large data sets, what you see on the left, and meaningful interpretation, which biologists and clinicians need on the right. So what you can see on the left, there is a variety of technology being available to us due to tremendous progress in sequencing and other technology. And now we can routinely study the genome, epigenome, transcriptome, and other aspects of the human body. But what is common between those technologies, they are all producing the data which is quite noisy and large, right? So there is two aspects here. So data is quite complex, requiring sophisticated and robust bioinformatics algorithm. So I'm excited about where the field is going, where bioinformatics is taking the central stage of the current biology, and we are moving to the data-centric world. So what that means that data plays a substantial role in many, many aspects of biology and immunology. And bioinformatics is the center of that. So the success of the project and success of discoveries often depends on the ability to do good bioinformatics. Oftentimes, the omics technologies are quite routine, and many companies are providing the service to sequence the human immunome or genome or whatever you would like to do. The bioinformatics is still tricky to be done. It's not straightforward. So there are many aspects of that. So spending time there, perhaps it's a good investment to ensure the project moves forward. But it comes with the challenges, and challenges are the big data. The data is really large and requires sophisticated tools to be developed. Big data management, so we need to manage the data. We need tools which are accurate, robust, scalable, and we need good databases. There is some ethical concerns. Perhaps you're hearing about AI and ChatGPT. The same in bioinformatics, we have quite a bit of that. Data access and resources, we need resources to run the analysis. So if you would like, we recently published a paper in Cell, kind of expressing our opinion of the field, so that you can see the link there. I think if you just Google the name of the paper, I think you're going to find it, or just send me email. I'm happy to send you the copy. So, but the key of that, I think, which makes many labs like the one I'm running independent and positioned to make important discoveries, secondary analysis. So what do we mean by that? So to put it in the simple terms, if you think about typical analysis performed in the wet lab, you would make those steps, what you can see here, where you would start with the biological samples, which you need to recruit the patients or obtain the samples anyhow else. You would do some sort of technology like sequencing. You would run computational analysis, and you will arrive, hopefully, at important results. During the process, luckily for us, who is on that side doing bioinformatics, nowadays you are required to share the data. Oftentimes you cannot publish the paper if you don't share the data. So what that means, every study in the world which is publishing those results will share the data in the public domain. So what that means? So we have data around which we can use right away. We can skip the first part, which is often costly, laborious, and we can download the existing data and try to look again at it. Try to do what we call secondary analysis, hoping that we can make as important discoveries as initially was done. So the question is like, why do we hope for that? Like, why would you look at the same data again? And there are many reasons for that. One, you can just do better bioinformatics. You can do better analysis, which can result in a discovery. Or perhaps the most common way, you would do meta-analysis, meaning that you would combine multiple studies which exist, and you would try to find something which initially was not found per study. Because let's say that study did lack the adequate sample size. But now agglomerating the studies together, you might have that sample size needed for that significant p-value to report. Or what I will be talking about today, you can what we call repurpose the technology. You can look differently at the data. So the way I would encourage you to think about that, it's one data set's many discoveries, so many ways to analyze the data. It's not exhausted. Like it's not one way to do that. There are multiple ways you can do it. So that fact that every study shares the data resulted that we have quite a bit of data sets available to you as we speak. Like literally all you need to do to go to SRA, Sequencing Read Archive, or GEO, Gene Expression Omnibus, and you will find a variety of the data sets. Unfortunately, not of them are publicly available. Some of them you would need to require the access to. And it's an interesting question, like what is the correct way to do it? Like, are we supposed to share every single data set? Are we supposed to hold it back? I think it depends on the nation. So for you guys, I think you should be proud being in Ukraine, because Ukraine national project was shared publicly. So there are 100 individuals from Ukraine, and then all of them, of course, it's anonymized, so you don't know who are those people, but the data is public, sitting on SRA, which is, for example, compared to UK Biobank, which is the leading project in the world, is restricted. You need to apply for the access. Okay, so one thing to realize, so data availability is not data reuse, meaning that you need to do more than just dump the data on the public domain. You need to do something more, like, for example, prepare good metadata, things like that. So if you're curious about, and I think I will almost done with the introduction part. If you're curious about that data reuse, and I think the way I would encourage you to think, I'm always curious about things, and sometimes if I'm curious enough, I just do them. So in this case, we were curious, like what is the fraction of the papers worldwide doing secondary analysis, like reuse, versus primary analysis, like traditional wet lab research? If you have the question like that, I would encourage you to be curious enough to do the analysis. And this was the case for us. We said, let's just figure it out. We want to know the answer. The answer is in the data. And this is, I think, to me, my favorite part about bioinformatics. Many things you would like to know, they are in the data. It's a matter of looking inside and knowing how to do that and developing the right algorithm. So what we did, we took 5 million publications across the globe. We download the data, look inside, and I can talk more about that. We designed the procedure to say, oh, that's a secondary analysis than the primary, and we look around. We look across the years. So I think what you can see on the left, on the plot, I think it's tremendous. It's very interesting. So you can see that we were uploading the data on the geo from early 2000, but we didn't have much data, as you can see. As we got more and more data, secondary analysis was not a thing. It was not popular. But as the time went, it is more popular scientific approach than the primary analysis. In other words, we're publishing more papers doing secondary analysis than the primary. So that means something. So that means that people are using that. It's a powerful tool. So if that was gene expression data, sorry, I forgot to mention. So here, the next plot is the raw data. So raw data is very hard to reuse. You need to know to rerun the analysis and so on. So that's not there yet. The numbers of reuse is not that high. Yet we are not using all the data. There's a lot of data sets never being touched again, meaning it was generated, it was uploaded, but nobody bothered to look again. Why is that? We don't know, right? Like we perhaps don't have the critical mass of people doing that. But that's important to know because there is a quite tremendous spending. There is quite big money being involved to make the data available to you, run by NCBI, and NIH, National Institute of Science in US, where there is big money spent to make that infrastructure available, yet we don't take advantage of that. But there are some tremendous data sets, and I provide, and this is what you can see, a couple of examples of that. So there are data sets being reused 15,000 times. So it's not just reused, it's been successfully reused, meaning there are 15,000 publications reusing the same data. So there is some tremendous potential of the data. And I think that the way I would like you to think about that is what is more important, what you do with your data or what the community does with your data? Right? So in this case, perhaps I would argue that it's more important what people have done with the data, because somehow they found it extremely, extremely useful. And I think the overall trends are interesting. I think something happened during the COVID. People perhaps were not able to generate some data sets. They couldn't go to the wet labs, so they were forced into some degree to reuse the data. So you can see some spikes. Or maybe it was some crucial number of people doing that. So, but the trends are there, and we were learning the trends and trying to make sense of them. Okay, so I think to wrap it up with that introduction part, right? So that was about bioinformatics. So now we can move to adaptive immune repertoire and how we can apply bioinformatics here in this particular case. So just to give you some background, so that some of you perhaps know. So if we talk about the adaptive immune repertoire, we can call it AIR with double R. And so that's encoded by multiple loci located across your genome. And we will be talking about Ig, immunoglobulin, and TCR, or in other words, BCR, B-cell receptor repertoire, and the TCR, T-cell receptor repertoire. And there are multiple chains there. So there is alpha chain and beta chain for TCR, for example, and then heavy chain and light chains for the Ig, for the immunoglobulin. But if you zoom in one of those loci, what you can see that there are a variety of V genes, which stands for variable, D genes, which stands for diversity, and J genes, which stands for joining. But there is not so many of them. There is like a decent number of them. So what is happening, they can be recombined in the random fashion. So for example, what you can see here, we are recombining V1, D3, and J2, right? So we're recombining them. And there is a constant region. But perhaps if you think about that and the way we on the bioinformatics side think about that, that's a combinatorial diversity, meaning how many combinations that can be calculated. But the number will be not that large because the number of genes is not large. But perhaps you heard that both TCR and BCR has astonishing diversity. Like in our body, we have 10 into the power 15 distinct sequences. I mean, immunogenomic sequencing being there. So that machinery is perhaps not enough. So what else we can do? We can insert non-template insertions at the junction points between V and D and D and J. So that machinery can generate literally any sequence. And this is the reason behind the diversity of the immune repertoire is so high. So we'll be talking about a couple of definitions: CDR free, complementary determining region, number 1, 2, and 3. And number 3, CDR free, complementary determining region number 3, is the most interesting because it kind of captures the most diversity. You can look there and perhaps this is where the most diversity will be happening. So we're going to be using CDR free sequence as the proxy for the clonal.